Good morning, good afternoon, wherever you are. Thank you for joining us today and for the next 3 days for the Brain Cell Atlas from Data to Knowledge workshop. I'm Andrea Beckel-Mitchener. I'm currently the acting director of NIMH, National Institute of Mental Health, and I am the deputy director for the Brain Initiative. So I get the fun job of welcoming everybody and opening up the first session. I just want to go back a little bit to 2014 when we first made awards on the very first phase of this project. We basically had a hope and an ambitious vision. I don't think any of us that fall of 2014 would have imagined what this project could become. And for me personally, this has been one of the most fun and scientifically, intellectually stimulating activities and rewarding that I've been involved in, having come to public service many years ago at NIMH. So it's a real pleasure to be here for this workshop. I am going to hand it over to the director of the Brain Initiative, John Ngai. John.
Thanks, Andrea, and welcome everybody. Thanks to Yong and Laura for organizing this 3-day workshop, to all the speakers, panelists, and all the participants for your engagement on this really important and timely topic. Also, I'd like to thank our colleagues at Ripple Effect for their excellent technical support. So as Andrea mentioned, the Brain Initiative Cell Census Project is now entering its 12th year. It started in 2024 when Andrea concocted this, what we call the Brain Initiative Cell Census Consortium. It wound up being 12 pilot projects that were stood up to develop and validate scalable technologies for creating brain cell atlases. And the effort in turn was scaled up in 2017. We were kind of pushed to do that. And that effort became the Brain Initiative Cell Census Network, or BICCN, whose charge was to deliver a comprehensive cell census, an atlas of the whole mouse brain, and draft cell atlases of non-human primate and human brains, with the idea of once we have the ability to do that, we can scale up to full big brains. I think it's fair to say that the BICCN has met and even exceeded all of these goals, and it's resulted in 4 major publication packages, numerous experimental and computational resources that frankly didn't exist when the project started, and really importantly, a growing trove of data that will enable new ways of studying the brain over the lifespan, and very importantly, in both health and disease. So now we're in the 3rd phase of the project, the Brain Initiative Cell Atlas Network, or BICAN, which is now focused on generating, among other things, a cell atlas of the entire human brain, as well as to discover the variation and the reasons behind the variation of cells between individuals. So, as Andrea mentioned, the progress of the Brain Cell Census Project over the past decade or so has been staggering. I just can't find another word to describe it. In terms of data generation, we've shattered Moore's law and moreover have developed ways to obtain and integrate data from multiple modalities, including single-cell transcriptomics, epigenomics, spatial transcriptomics, and now expanding efforts in proteomics. So it's really been quite exciting with a lot more yet to come. But the big challenge facing us now is turning this growing mountain of data into knowledge, and in a form that can be democratized and accessed by researchers around the world to make new discoveries about the cellular basis of neural circuit and brain function, and also to identify the causal basis of neurologic and neuropsychiatric disorders that affect about 1 in 3 humans on the planet, and ultimately pave the way toward new cures. So I'm really looking forward to the next 3 days as we evaluate the current state of the art and discuss the challenges and opportunities for developing a robust brain knowledge base. So with that, I'm going to turn it over to Yong for a more comprehensive introduction. Yong?
Thanks, John. My name is Yong. I'm Program Officer of the NIH Brain Initiative and the National Institute of Mental Health. I would like to start with providing a background and context of this Brain Cell Atlas workshop. As John just mentioned, the Brain Initiative Cell Atlas Network, BICAN Consortium, was launched in 2022, following a successful completion of 3-year pilot and 5-year brain cell census projects. The network has currently over 500 members and affiliate members, who are mainly from 5 data generation centers, a group of complementary co-laboratories, and technology development projects, 4 data coordinating projects, 5 Brain Initiative data archives to disseminate the large volume data generated by network, a brain specimens core, and 2 gene sequencing centers. The overarching goal of BICAN is to create comprehensive and spatially resolved reference brain cell atlases in human and other mammalian species as a foundational framework for the study of brain function and disorders. A variety of assay technologies are adopted by BICAN to create brain cell atlases, as you can see from this slide, including single-cell genomics, spatial transcriptomics, brain histology, MRI imaging, and brain cell functional recordings. In particular, high-throughput single-cell transcriptomics and epigenomics and spatial transcriptomics technologies have enabled a large-scale characterization of the cell diversity across human and mammalian species and age groups. A large number of single-cell genomics profiles in the order of 100 millions will be generated and analyzed by the consortium. And about 1 billion brain cells will be characterized by spatial transcriptomics methods. More recently, a center was founded to establish a 3D cellular imaging platform to characterize protein and RNA expression in high throughput manner. If successful, about 1 trillion of brain cells will be profiled in whole human and non-human primate brains. And the raw data size of BICAN will amount to about 100 petabytes. BICAN consortium is now in the process of submitting the first round of joint publication package of over 40 papers focusing on basal ganglia in human, macaque, and marmoset and mouse. These papers will present a cross-species consensus atlas of the basal ganglia, epigenetic regulation underlying cell type specific gene expression, inter-individual variation, morphological and electrophysiological diversity, genetic tools, as well as the development of basal ganglia. In addition, the joint publications will present how large-scale multimodal data are managed through coordinated consortium-wide efforts in BICAN. There are also efforts to map cellular gene expression according to harmonized brain anatomical ontology and the brain common coordinate frameworks, thereby bridging genomics to brain connectomics. As an example, one of the joint publications by Xiaoming Xu lab in UCI in collaboration with Bogdan Bintu lab at UCSD showed that the Murphy plus imaging-based spatial transcriptomics method enables the highly precise localization of individual RNA molecules to individual cellular compartments. Each RNA transcript can be categorized as nuclear, somatic within the cytoplasm, or putative cellular processes, including neurites, to provide information about neuronal connectivity. They found, consistent with the basal ganglia circuitry found in the mouse and monkey model organisms, that D1 and D2 medium spiny neuron enriched transcripts exhibit higher extrasomatic signal density within the respective projection target regions in human brain. Thus, their preliminary data suggests the precise spatial location and quantitative correlation analysis of neuron synaptic and somatic transcriptomes may offer a new way to infer neuronal short and long distance connectivities in human brain. The joint publications will describe how the brain cell atlas data will be managed and disseminated by the BICAN data ecosystem. There are still challenges in integrating the diverse brain cell properties and phenotypes collected by BICAN and the broad research community. In particular, it remains unclear how will multiple layers of data sets and annotations stored in different data entities be linked in brain cell atlas, and how will the vast spatial scales and heterogeneous data modalities be integrated, and how will the intersubject variabilities and the intrasubject dynamic changes will be characterized. This three-day workshop will tackle these challenges and develop a roadmap, hopefully, for establishing a comprehensive brain cell atlas data to knowledge pipeline in future. There will be 2 panel discussions every day following keynote presentations. The objectives of day 1 on data annotation and integration is to establish unified brain cell taxonomies and the reference brain cell atlases by leveraging AI machine learning techniques. The objective of day 2 on data commons and AI-powered knowledge bases is to develop a robust data infrastructure and automated pipelines for data annotation and knowledge derivation. The objective of day 3, impact on brain function and disease research, is to explore how the brain cell atlases can provide the new insights into brain function, cellular connectivity, and disease mechanism studies. The final plenary session will hopefully present a roadmap toward a unified and federated brain knowledge base framework. I would like to put a plug for the brain knowledge base innovation domain that is a new strategic ambition of the NIH Brain Initiative to address emerging data management challenges. The envisioned brain knowledge base framework will leverage brain's domain-specific expertise and consortium-scale data platforms to enable transdisciplinary advances across the spectrum of neuroscience, from molecular mechanism to cognition and behavior in a sustainable and ethical manner. The brain knowledge base framework will include a federation of specialized brain data archives and a federation of specialized knowledge bases, including brain cell atlas knowledge base, brain connectivity across scales connects knowledge base, and the brain behavior quantification and synchronization BBQS knowledge base to manage data to knowledge pipelines across molecular, cellular, circuit system, and functional behavior dimensions. I'd like to thank you all for joining this exciting workshop. I look forward to the success of this workshop.